अज्ञानतिमिरान्धस्य ज्ञानाञ्जनशलाकया चक्षुरुन्मिलितं येन तस्मै श्रीगुरवे नमः. सर्वेभ्यः नमो नमः. Very good afternoon to all of you. Respected expert panelists, today's presenter, teaching faculty, PhD and PG scholars from various healthcare institutes across India. I, Dr. Vinay Pawar, Department of Sanskrit Samhita Siddhanta, D.Y. Patil, deemed to be University School of Ayurveda, Navi Mumbai, on behalf of JIM Journal Club Organizing Committee, welcome you all for the 17th online JIM Journal Club. Dr. Arti Chandran, final year PG scholar, Department of Rasashastra and Bhavishya Kalpana, Paril Institute of Ayurveda, Paril University, Gujarat, is going to present published paper entitled Swarnamakshik Bhasma Preparation using an alternative heating method to traditional Varaha food. We are fortunate to have two eminent personalities as an expert panelist for today's journal club. Professor Avinash Kumbhar sir, Head, Department of Chemistry, Savitribai Phule, Pune University, Pune, and Professor Asmita Ville Madam, Head, Department of Rasashastra, Bharati Vidyapit, DIMB University, Pune. We heartily welcome you both. With the permission of expert panelists, I would request Dr. Arti Chandran to begin with her presentation. Dr. Arti Chandran. Dr. Arti, can you share your PPTs? Arti, are you there? Okay. Okay. Can you make it full screen? Arti, you are not audible. Arti, is there any technical issue on your side? Make it full screen. Is it possible?
Arti, you are not audible. Hello.
Arti, are you there?
Is it visible?
It's visible, but not full screen. No, I think there is some problem because when I tried the same, but it's not coming full screen presentation. I don't know. Can you increase the size? Make it large. Hello? It's not readable actually at present. By the time the participants can go through the paper, I have shared link in the chat box. They can go through the paper. To invite participation, Arti, are you ready? Hello? Arti? Okay, okay. Begin with your presentation.
Hello, sir. Am I audible?
Yes.
Thank you, sir, and sorry for the inconveniences.
Okay, no problem.
I'm very grateful to be part of this JIM Journal Club, and I would like to thank all the JIM members and authors of the article for their contribution and all our faculties.
Yes, Arti, please proceed.
Please go ahead. Yeah.
The slide is not changing.
Hello. What happened, Arti?
Sir, slide is not getting changed.
What?
Slide change.
Okay. From your side, what happened?
Please email slide to
CCIH.
CCIH.
Okay, sir.
Arti, you don't do anything. Vinay will message.
Okay.
Arti, you please start.
You just keep on talking
You
about
continue.
your
We'll
paper.
take care of slides, Arti. Don't worry. Stop sharing your screen, Arti. And please start.
So please check your inbox.
Yes, Arti, please start. We will share screen. Please continue.
Okay, sir. For this journal club, we have chosen one article from JIM journal, Swarnamakshika Bhasma preparation using an alternative heating method to traditional Varahapuda. The authors are PMYS Patiraja, YMMK Rana Tunga, etc. Except respected YMMK Rana Tunga, all other authors are from engineering field, and YMMK Rana Tunga from Institute of Indigenous Medicine, Colombia, Sri Lanka. Abstract. Background of the study is Ayurvedic system of medicine have abundant formulations. While during the preparations, the mineral will change into the form. We used to change the form of minerals into bhasma. So number of pharmaceutical procedures included under Bhasmikarana process. So the main concept of the study is that, and the objective of the study is to obtain the temperature profile of the traditional Varahapuda and to establish a complementary temperature profile in a muffle furnace. Introduction at a glance, they have explained the definition of Bhasma, Shodhana, Bhavana, Marana, Puda, and importance of temperature in Bhasmikarana. Materials, unavailability of the raw drug Swarnamakshiga in Sri Lanka, they have imported Swarnamakshiga from Indian market, and remaining all the materials are collected from local market of Sri Lanka. Method, analysis of Swarnamakshika, Shodhana of Swarnamakshika Bhasma, Shodhana of Gandhaka, preparation of Vanopal and evaluation of calorific value, Marana of Swarnamakshika, analysis of Swarnamakshika Bhasma. They have analyzed the components of Swarnamakshika with the help of atomic absorption spectrometer. And they find out the percentage of copper in the raw materials are very less as compared to the standard proportion. And they have find out percentage of silica in the raw material also. It is the standard proportion of chalcopyrite, that is CuFeS2. They have considered it as the standard one. 30.5% of copper, 30.5% of iron, and 35% of sulfur by weight. To maintain this proportion, they have added 95 grams of copper sulfide in 90 grams of the raw material, and the total mixture of this two is considered as Swarnamakshiga for the study. Shodhana of raw materials. First, purification of Swarnamakshiga. They have to reference from Rasa Tarangini 21st Taranga. And they have taken 180 grams of Ashodhita Swarnamakshiga mixture and added 200 ml of Nimbu Swarasa in that and roasted in a Loha Kadai for 48 minutes. And Gandhaga purification. For Marana process of Swarnamakshiga, Gandhaga is needed. And they have followed Rasa Tarangini 8th Taranga second method, that is Gandhaga Mani preparation. Equally dimensioned two pot were taken, and the volume of the pot was about 2 liters. And in one pot, they have poured 1 liter of cow's milk and added 245 grams of cow's ghee in that. And the mouth is covered with cotton cloth and tied it well, and then applied the powdered Ashodhita Gandhaga about 245 grams over the cloth, and the second pot is placed over the mud, the first pot, and then Sandhibandhana, and then they followed the Adapadana method and used 20 number of cow dung cakes for the procedure. And then they have prepared Vanopalas, cow dung cakes. They have tried to optimize the proportion of the preparation of cow dung cakes. They have chosen 4 proportions, and for first 3 methods, they have only included cow dung and paddy husk. For the 4th one, they have added sawdust also in it. They prepared the cow dung cakes with the diameter of 6 inches of diameter and 1 mm, 1 inch in thickness. And they calculated the calorific value. The calorific value is the energy which contained by a fuel or food, which is calculated with the help of bone calorimeter. And they got most calorific value, that is 15.44 megajoules per kilogram in the proportion of 76 parts of cow dung and 24 parts of paddy husk. And the moisture content is probably less in that, and this composition is considered for the full study. Pre-preparation for Marana of Swarnamakshiga. And the Shodhita Swarnamakshiga and Shodhita Gandhaga were taken in equal amount. Here they have taken 200 grams of each the ingredients and triturated with sufficient quantity of Nimbu Swarasa. And the reference is taken from Rasaratna Samuchaya second chapter, and the same reference is available in Rasa Tarangini 21st Taranga. And after proper trituration, they have prepared pellets having 2 centimeter in diameter and 3 mm in thickness. After the drying of pellets, they have done Sabbudhikarana. And then the process of Marana, they have chosen 2 methods, Varaha Puda and muffle furnace. For the calculating the temperature of Varaha Puda, they have used thermo data logger to calculate the temperature. Then first Varaha Puda method. For Varaha Puda, they have dig a pit having the dimension of 1 arachne height, length and width and depth, and it is about 42 centimeter in SI units, and they have filled 80 cow dung cakes first in the pit and placed the Sampudita Sharava over it, and then for remaining 40 cow dung cakes are filled with the Varahapuda and ignited from the four sides. And the temperature profile was recorded, and then they have go for the muffle furnace. The same temperature profile was followed in muffle furnace. After the first cycle of this Swarnamakshiga Bhasma Pudana, they have collected the Pudita Dravya and do Bhavana with Nimbu Swarasa and continue the same process for 4 Pudas, and they obtain the Swarnamakshiga Bhasma. It is the temperature profile of Varaha Puda and muffle furnace. After ignition, the temperature rises after 35 minutes and reach the peak value after 1 hour and 40 minutes. After combustion, it will take 3 hours to reach the normal temperature in Varaha Puda. The same pattern is followed in muffle furnace also, but the time taken for the condensation is more in muffle furnace. It was taken about 6 hours. The difference is obtained because of the qualities of the system. Because in Varahapuda, it is just connected with atmospheric temperature, and it is a multi-thermal system. And in muffle furnace, it is closed and the heat distribution is or equal in all the walls. So the condensation will take more time to come to normal temperature. And then observations of the incineration process. Around 12.5 to 12.9 kilograms of cow dung cakes were used for Pudana, and the number of pellets of Varaha Puda and muffle furnace are same, showing the same changes also. But in the second Puda, we can observe the drastic change in the quantity of the pellets. It is because in first Puda, they have added equal quantity of Swarnamakshika and equal quantity of Gandhaka in it, and then they have prepared the pellets. From the second Puda, they didn't add the Gandhaka in that. So that's why the drastic change of the weight of first Puda and second Puda were occurred. And examination of the Bhasma, they have examined in 2 ways in Bhasma Pariksha laboratory investigations. In Bhasma Pariksha, they have done Rekha Puranatva, Varitaratva and Niruddha. Laboratory investigations, organoleptic characters, total ash, acid insoluble ash, loss on drying, particle size distribution, and chemical composition. Color of the resultant Bhasma, it is blackish. In the first Puda, it was about blackish, and after 5th Puda, they got a brownish red colored Swarnamakshiga Bhasma in both muffle furnace and Varaha Puda. And the final Bhasma properties, color is reddish brown in both muffle furnace and Varaha Puda. Ekha Puranatva, Varitaratva, Niruddha were satisfied in both muffle furnace and Varaha Puda. And loss on drying, the loss on drying, the standard, the Swarnamakshika Bhasma have the standard, satisfies the standard values, but little more in Varaha Puda. It is because of atmospheric changes. And then total ash, the total ash will determine the quality of the Bhasma. The higher total ash value proves the high quality of inorganic substances in the Bhasma. And acid insoluble ash, it is doing to analyze the siliceous materials of the Bhasma. It is also satisfies both the muffle furnace and Varaha Puda procedure. And particle size distribution of both final products. The particle size distribution test was done with laser particle size analyzer, and the 0.5 to 92.5 micrometer in diameter is the average size of the Swarnamakshiga Bhasma, and more 10 micrometer in diameter particles are more found in muffle furnace because of the uniform heat, and 20 micrometer in diameter is same in both of the system. And chemical composition of Swarnamakshiga and Swarnamakshiga Bhasma was again analyzed with atomic absorption spectrometer. And the raw drug, the percentage of raw drug is just half to after the preparation of pellets. It's because of they have added Gandhaga with that, and then they have did the study. So the composition or components are less in pellets. And in final Bhasma, Varaha Puda and muffle furnace, the drastic change and high values of the components are seen here. It is because of the artificially prepared Swarnamakshiga is used for the study. And conclusion, the 76% of cow dung and 24% of paddy husk proportion of Vanopala got more calorific value, that is 15.44 megajoules per kilograms. And the temperature profile of both Varaha Puda is continued for the muffle furnace, and the result is same as that of Varaha Puda, muffle furnace and Varaha Puda. So they concluded that both Varaha Puda and muffle furnace, we can compare the studies, and we can continue, we can use instead of muffle furnace instead of Varaha Puda. And presenter's view, calculation of calorific value, it is a very advantage step about Ayurveda. They have calculated the calorific value of Vanopala, and they have optimized the proportion of Vanopalas. And Bhavana is also considered as one of the Shodhana, but they have explained it as different procedures in the introduction and abstract. And external addition of copper sulfide. They have added externally copper sulfide to maintain the percentage of copper in the Swarnamakshiga Bhasma. In our classics, no such references are found, and they didn't explain the method of adding copper sulfate in the Swarnamakshiga Bhasma. And Shodhana of Swarnamakshiga, they have quoted that the method of Shodhana is taken from Rasa Vagbada Acharya Rasa Ratna Samshaya, but the actual reference is from Rasa Tarangini 21st Taranga. And they have done the Shodhana for only 48 minutes, and they didn't recorded the observations, but in our classical reference, they have clearly mentioned that we have to heat it for more time. It was about 2 to 3 days. And accuracy in quantity of raw drugs, they have explained the different quantity in different sentences or different headings under different headings. First of all, they have took 90 grams of Swarnamakshiga with 95 grams of copper sulfide. So it is total 185 grams of raw drug, and they have took 180 for the Shodhana, and for the preparation of pellets and for Marana, they have took 200 grams of Swarnamakshiga. And Marana process, in the reference, they have explained that the whole process have to repeat it for 5 times. The whole process means equal quantity of Swarnamakshiga and Gandhaga was triturated with Nimbu Swarasa, and then repeat the process after Pudas. But in this article, they haven't added Gandhaga from the second Pudana process. And Bhasma Parikshas, they have did only Varitaratva, Rekhapuranatva and Niruddha. But Swarnamakshiga Bhasma is having the main constituent of as copper. So we have to do Avami, Amla Pariksha and Nisadu Pariksha for to confirm the proper formation of Bhasma. And Nishchandratva is also indicated for Swarnamakshiga Bhasma Pariksha. And before going to the next point, I would like to explain one article. Sudhal Dev Mohapatra et al. Have done a study, pharmaceutical standardization analytical study of raw Swarnamakshiga and its Bhasma through the TM and EDAX. So they have recorded components of Swarnamakshiga are copper 33.08%, iron 28.60%, sulfur 32.85%, oxygen 5.47%, and after Shodhana, the percentage of the constituents are copper 21.45%, iron 49.25%, sulfur 0.838%, and oxygen 20.92% weight by weight. And the Swarnamakshiga Bhasma is having copper, iron, sulfur, oxygen, magnesium, aluminium, silica, and potassium as the components. And this is also one more study. It also reviews the same compositions. And here, constituents of raw material, raw Swarnamakshiga, they have included silica as a component, but they didn't mention sulfur as a component. And they have added copper sulfate extra to the raw material to make the copper percentage also. Then also they didn't explain about sulfur percentage. And it is the proof for adding copper sulfate to the Swarnamakshiga. And constituents of Swarnamakshiga Bhasma, they have done, it's true, atomic absorption spectrometer only, but they have explained it as in a compound form here. And here also they have did the same, the AAS examination only, and they got a very less amount of copper in that. So, and then the practical view of Swarnamakshiga, they have used 120 cow dung cakes and the weighs about 12.5 to 12.9 kilograms, and they got the Bhasma within 5 pudas. But they didn't follow the classical method, but they got the Bhasma within 5 pudas. But here one article I'm quoting, it is also by Mohapatra et al. And they have did the study with 4 kilograms of cow dung cakes, and they got the Bhasma with after 9 pudas, and here they have added Gandhaka also in it. So it's one of my query, the standard procedure for Swarnamakshiga Bhasmikarana. And in summary, preparation of Manopala and the calculation of calorific value is clear through this article. The optimization of Manopala is also cleared about the calorific value of the Vanopala. And preparation of Chakrika, they also standardized the preparation of Chakrika, and we got clear knowledge about that also. And importance of heat in Bhasmikarana, the same here they have used very high quantum of heat in the Bhasmikarana process, and they got very easily in the Bhasmas within 5 pudas. And then chemistry behind the Pudana process, the number of chemical and physical changes will occur in the Pudana process, and the resultant product will shows the effect of the chemical and physical changes which are happen in Bhasmikarana process. And the classical and muffle furnace have some similarities because they are using for to obtain high temperature, and the increase and decrease of temperature is very step by step, and the substance is isolated in Parahapuda and in muffle furnace. And these are all the references. And I would like to acknowledge to the organization members of JAIM, our respected Dean, PIA Parul University, all the RSBK faculties of Parul University, Dr. Rekhi sir, Professor, RSBK Department, Ashwini Ayurveda College, Davangere, Dr. Mahesh sir, Research Officer, Central Ayurveda Research Institute, CCRAS, Patiala, Punjab, authors of the articles, and all my colleagues for providing such an opportunity and support. Thank you.
Okay. Thank you, Aarti, and congratulations for presenting a paper. Very good paper. Very good presentation of the paper with critical comments. And you have also done comparison with this paper, comparison with other published articles and classical texts also. So it was a very good attempt. Thanks for that and congratulations. I would request our expert panelists to say a few words or to give their critical comments on this paper and the presentation also. Would request Professor Avinash Kumbhar sir to give his critical comments on this.
Thanks, sir.
Your voice is breaking, sir.
Hello.
Yeah.
Am I audible?
Yeah.
Hello.
Yes, sir. Yes.
No, I was just wondering, should we ask Asmita madam to go first?
Okay. Okay.
Yes,
Okay.
yes, yes.
If she, yeah.
I would request Dr. Asmita Vele madam to give critical comments on this paper and the presentation. Asmita Vele madam.
So, thank you, the CCIH team. Hello, Dr. Kumbhar sir,
Hello.
Girish, Vinay, and the presenter. Indeed, this was a good paper, and Dr. Aarti Agama presented it well. What I liked about her is she has also checked for similar research works, especially done on this Swarnamakshik Bhasma. She has compared it with the classical texts as well as the other texts. What, when such papers and it's a small paper or a research work is a small part of the whole big knowledge system. And in that sense, for this particular paper, they have stuck to a manufacturing process which is classically conducted for Swarnamakshik Bhasma, and the same process conducted using advanced manufacturing technique, that is muffle furnace. So I think that in that perspective, what they have done is they have achieved their objective, which was there. But at the same time, the raw material which they have procured, it is Swarnamakshik. I read in the comment in the chat box whether it is Swarnamakshik, it looks like Vimal. I can show you a couple of samples. I don't know whether I can share the screen. Yeah, there is a possibility of sharing screen. Quickly, I'll show you the samples. This is there. Share screen. Should I use that?
Yes,
Share screen. Desktop
Yes,
one.
madam. Yes.
Share. Give me a moment.
Yeah.
Is it visible?
Yeah. Yes, madam.
Okay. So there are a few samples here, right? This sample is also Swarnamakshik. This one, the first one. I think it's, my cursor is moving. Yes. The second one is also Swarnamakshik. So there are these two samples are definitely there. The third sample is also Suvarnamakshik. The amount of Suvarnamakshik in it is very small. The fourth sample, which is it's hidden here, but is it visible to all of you?
Yes.
In the same row,
Yeah.
the fourth sample,
Yes, madam.
that is not Suvarnamakshik. The fifth sample also has down below. It also has a limited amount of Suvarnamakshik. So yes, there are, this is a mineral. It is sourced from natural origin, it comes from mines, and therefore it might vary in its appearance. So there is a possibility that the sample does not contain, and Suvarnamakshik is also there in pharmacopoeia. So pharmacopoeial standards are there. And these people, the researchers, they have added CuSO4 to complement the content of Cu, is because of that pharmacopoeial standards, which is highly possible. Now, should we do it or we should not do it? This CuSO4, was it chemical, was it natural? If you ask me, I would go ahead with, I mean, there is no problem to add certain thing, but if we are talking about natural elements, then the sample should have been the right sample to start with, because CuSO4 chemically prepared copper and sulfuric acid reaction, that will be a completely different chemical moiety. Dr. Kumbhar, you might throw light on this particular point, and then we will go ahead so that the panelists will understand better. I mean, the participants.
Yes, rightly said, madam. Thank you, CCIH group, especially Tillu sir and his team for giving me the opportunity to be present on this paper presentation, and I thank Dr. Aarti for the presentation. I have also gone through this paper with great interest. The reason is that obviously it deals with the basic chemical, that is CuFeS2, as Madam had pointed. And in fact, also use this, we prefer to call it as chalcopyrite rather than Swarnamakshika, as is mentioned over here, because that's the ore or the mineral from which copper is extracted. So we use it as an analysis as an MSc practical for analytical to extract sample by simple titrametric methods or simple methods of decomposition, and then followed by its analysis by titration. And we can extend it to other copper minerals, including some alloys such as brass. So we have a method to analyze that. And as was mentioned over here, there is a comparison between the traditional method in terms of heating with the I would say an artificial method of heating by using a muffle furnace, and the composition was found to be similar, and so therefore they said that this can be used. I had one couple of points that I would like to mention. One is the reproducibility in the formation of this product, both by the traditional method as well as by the muffle furnace. So how reproducible it is in terms of the constituents. That is one thing that I'm sure the authors must have taken care of, but in general, we as chemists want to know how reproducible is the system. Next is the sequence of addition of the ingredients. What was the sequence of addition of the ingredients? Was there any change, or has that been kept constant? That is one aspect that we want to, I mean, I'm sure they must have taken care of, but this is what we as chemists take care of, because we know that the local environment changes drastically because of the change in the sequence of the constituents or addition of the constituents. Another aspect, yes, the temperature that is mentioned here, roughly the temperature goes to, I think, around 800 degrees centigrade, 800 to 900 degrees centigrade. So it is not surprising that the muffle furnace ingredients as well as the traditional ingredients seem to be the same at around that temperature. However, uniformity may be better in the case of the muffle furnace, but if the methods have been standardized for the traditional methods, I see there is no problem in the constituents. And chemically CuFeS2, we would obviously give more importance to the copper and the iron, which are the metals that are present over there. But actually, sulfur plays a major role in connecting or contacting these two metals. And in fact, at a higher temperature, about 1000, the composition changes. The CuFeS2 that we see, that changes to some different composition. It's basically 5 Cu and FeS2 is what is formed at a higher temperature. That's the reason after 1000 degrees centigrade, maybe the constituents will go on changing because the structure goes on changing. Copper starts dominating, and copper, yes. Yes, please.
Yes, and that's why, sir, the temperatures while heating are always kept to a certain limited level. If this is Varahaputta, the temperature range in Varahaputta generally should not go beyond 900. And at the same time, the studies which we have done, and many people in Rasa Shastra, they have done in Jamnagar especially, lot of studies have been published. The temperature in Gajaputta goes between 900 to 1000.
Right.
And there are compound
changes,
minerals.
Correct.
So always compound minerals, just like this Abrak or Suvarnamakshik or Vimal, that is CuFeS2 or FeSO4. These are processed in that. Also, all the pure metals, chemically pure metals, which are processed using Ayurvedic techniques like Shodhan, Bhavana, and Putta, that is Maran. So all these 3 steps in the process, Shodhan, there are multiple processes, either quenching, dipping, or trituration, or size reduction using certain, addition of plant alkalis, salts, and then going ahead with the trituration, making pellets and heating in Putta. So for all the dhatus, the temperature range is used beyond 900, but not beyond 1000. So between 900 to 1000, 1050, like that. And when there are these compounds like CuFeS2 and others also, ZnSO4, ZnCO3, there we use Varahaputta generally for Suvarnamakshik, Vimal, Kharpar, and all others. So I think, and there are repetitions until the final product passes certain confirmatory tests. These tests, in this paper, these tests are clearly mentioned. There are some other tests also, and our presenter pointed it out very well that these 3 tests have not been done. I also saw one question: what was the amount for Niswadu test, or what was the amount for Vami? So that is a question. Some students should take up a small project on the just to identify how to standardize this Avami test for a Bhasma. Today we do not know what is the amount, how long we should check it, whether that sensation of nausea and vomiting that occurs in 15 minutes, 20 minutes, 1 hour, 2 hours, 24 hours. We do not know. So definitely there can be a small project, the Bhasmas, where Avami test is very specifically mentioned. I mean, for all the Bhasmas we can do it, but there are certain Bhasmas where it is very specifically mentioned, like copper, Tamra Bhasma, like this Suvarnamakshik Bhasma, and a few others. we can have a standardized method. This is for all the participants, and many of them are teachers, so they can take up this small project with one of their PG students, and we can go ahead on that. For Niswadu, I think a pinch is sufficient, and that is everywhere. You take a pinch, you put it on the tip of the tongue, and you taste it. Now, if we want to really check it, and we know that different types of tastes are perceived at different locations on the tongue. Going by our text, if a bhasma is tikta kashaya, if a bhasma is madhura kashaya, if a bhasma is madhura tikta, whatever is the taste, we should be able to again standardize this, on which part of the tongue that small amount, a pinch or a half of pinch amount of the bhasma should be put. So these two are new. As we were, I mean, this was a little diversion, a little moving ahead quickly.
Right.
But sir, as you were saying, the raw material here, my take-home is raw material should be characterized and tasted for one simple reason, that all these minerals and metals, they come from mines, and there are many associated materials along with this main material. So just like in herbs, we find different species in different in a genus. Similarly here, the veins in our mines are interlinked, and therefore mineralogists have gone ahead with the identification that if there is copper sulfate, if there is chalcopyrite, then arsenopyrite will also be there associated with it. Then iron pyrite will also be nearby. So we have to, when we get the mineral sourced, then there has to be a process of separation and collecting as far as possible the purest form of the mineral, then identifying it for particular marker compounds. Like here there are three: copper, iron and sulfur. That should be there, and then we should take it for further processing of Shodhan or Maran or whatever we want to do.
No, I'm okay. Let me also point out here is, I think I was really frightened by reading the first sentence of the paper in the introduction, which said that Ayurvedic medicinal powders prepared from minerals, metals, and poisonous materials. So I was wondering, gems combining with various herbs in the treatment. So when you mean the other minerals, it means these other poisonous material, which may also be present in trace quantities. So yes, in some cases, yes, they are also maybe required for this process. Obviously, the constituents may change later on, or the material will be maybe different. And another thing which I really was I don't know, a series of pharmaceutical processes. Yeah, processes.
Let me interrupt you one second. Here,
yes,
the meaning of the term poisonous materials is a class of drugs, which is called Visha Upavisha Varga. And the researcher has translated Sanskrit directly into English. And therefore, so it's poisonous materials means here there are 10 drugs in this class of poisonous poisonous starting materials or poisonous substances. We deal with, in Rasa Shastra, we deal with about 80 metals, minerals, and 10 poisonous herbal sources. And it's that. So Visha Upavisha here is nothing but, I mean, that's just an error of interpretation.
Right. And then I wanted another clarification from my part. Is that the bhasma undergoes a series of pharmaceutical processes, which you call as Shodhana, Bhavana, and Marana. My question was, pharmaceutical processes haven't yet started. I mean, it will come after your pellet formation and other. So how do we call these pharmaceutical? That's my question that I had. And just to stop, maybe to give, what is the final pharmaceutical effect that is expected of this bhasma? I think Sudeshna madam has said that it is used for anemia, or what, yeah. So,
So there are,
yeah.
yes, sir, pharmaceutical processes, as you know. Can you just guide this,
No,
all the participants how you look at pharmaceutical processes?
I look at pharmaceutical processes once this drug or medicine has been inserted into the biological system, has been given, has been taken by the biological system, or the humans, or animals in general. So that's the reason why I was saying pharmaceutical process, when you are saying in the making of the bhasma.
Yes, sir. Now what happens is pharmaceutical process starts from raw material. This is our the mineral which comes as it is our starting material. And the pharmaceutical processing is like size reduction, then heating it, trituration, converting it into bioassimilable form. Generally, the form is powder. The first form is powder. Then there are many other forms as well. But you transform them by mechanical heating as well as processing with different decoction, that is water in some form, liquid comes in, and then in some form heat is there. So with these two, water and heat, this raw material is converted into a palatable form, that is powder, and that's called bhasma. Now I am telling this to you with the basis of chemistry. All the Ayurvedic people will immediately start asking me, madam, we do not use water, madam, we use for a squash juice. Yes, I understand. I'm talking about the physical form, that there is this and there is heat. So different types of heats are there. Direct heating is there. In case of this particular drug, there are 4 different methods which are mentioned in classical texts, and people follow all these 4. So you heat it in open pan without putting anything. Just start heating it at a strong flame, and after the sulfur goes away, sulfur naturally, this is CuFeS2. From your viewpoint, there could be sulfur dioxide, but we crudely say that the fumes, they go away, and after that those fumes go away, we cover it, and then we heat it for about 3 days. Keep that constant heat on, and it is the charcoal heat, I mean the wooden heat. So this is a pharmaceutical process for us, and then after that we take it, then we triturate it with lemon juice. We also add some sulfur. There in other formulation there is hingur, that is cinnabar, which is processed with our ardraksvaras. That means gingiber officinalis juice. That is processed for 7 times. You add it, you dry it, then again add it, again dry it, again add it. So there are multi-layered processes, multi-step processes, and therefore these are all pharmaceutical processes. These are different. Yeah, I can just give me a second. I can see here the bhasma should be nisvadu. Yes, that's correct. Bhasma should not carry any taste, and similarly, there is also it should be nirgandha. So when you put it on, I mean, there should not be, when the bhasma is ready, there should not be any taste, there should not be any gandha. That's the optimum property that should be there. It is, every bhasma should pass to that taste. But yet I say that we have to, for certain tastes, for certain bhasmas, like calcium bhasmas, there is a taste that it should not burn your tongue. But we have to do a standardization of this. We should buy samples which are ready, available, follow a standard pattern, and then do it. And therefore there will be a standard. Otherwise, okay, we say, but yes, some there are some bhasmas which even I have tasted, and there are little taste to that, but they pass all other tastes of bhasma. In this case, what should we do? If you ask me, ideally, because I tasted a little taste of something in that bhasma, it should be rejected. because a bhasma should pass all the 10-15 tests which we expect. So that those are the I think research leads which we should do. Somebody has said bhasma itself is not a final medicinal thing. It is intermediate material to be used with herbs as per disease. See, there are different, I don't agree with this, because abhrag bhasma with madhu, suvarna bhasma with madhu, any bhasma with any of these three things, madhu, ghrita and taila, you can use as a final drug. Why can't you use it? It's not necessary that it should always be mixed with. These are the later on practices to get better benefits. People started using it with similar kind of herb, similar type of herb. So my basic question always, after working in this field for 30 years now, I feel that if there is a bhasma which you make using all these herbs, sometimes 15, 20, 30, 100 herbs, then why do you want to add one more herb to it? Just because it is a clinical practice? Do you really, I mean, do we really know what exactly is happening when this finished product bhasma goes inside our body? So that's what is the response to this particular comment. And sir, coming to your next point is used for anemia. Yes, Suvarna Makshit Bhasma. It's, if you look from the pharmacological viewpoint or the effect of a drug, then yes, there are three terms: Pandurog, Rudrog. Pandurog can be equated with anemia, okay, to certain extent, for a understanding, general understanding. Rudrog is cardiovascular diseases, and the third one is Rasayan. So we can, if you look from the Ayurvedic viewpoint, this is a very limited understanding. This particular drug can work in many diseases of Rasavaha Srotas, Raktavaha Srotas, both. I mean, it's kind of a broad spectrum drug. I'm not using the term antibiotic. It's a broad spectrum drug. There are many conditions with these two systems where this drug can work, and ultimately it is Rasayan. Now, Rasayan for what? My take after reading Gunesh Shastriji, Dhamankarji, and many others, especially the clinicians who have written these books, I feel, even Mama Shastri Gokhale, I feel that it works on these two diseases of these two systems, Rasavaha and Raktavaha. So we can explain it in some different, at some different time point. We can see research papers and all. That's what I feel. And now over to you, sir.
Avinash, sir.
I think he has left the meeting.
I think there could be some problem. I would, Girish, if you allow, I would like to tell the audience for their information that there are many papers on Suvarna Makshit Bhasma. The first initial papers you find is in 2007, where Lakshmi Prakash Gupta ji and C.B. Jhasan, they worked on some hypnotic and behavioral effect of Suvarna Makshit Bhasma. And there they, I think, they were questioned with the standardization and all. And then the other student of C.B. Jhasan, Vijay Lakshmi, R.K. Gupta, along with them, C.B. Jhasan conducted a trial where these 4 types of Suvarna Makshit Bhasma end products. Why I'm saying 4 types? The raw material is same, Suvarna Makshit, but the intermediate pharmaceutical ingredients change and process also changes. So in one final end product, there is lemon juice. The Shodhan was done by intense heating up to 900 degrees. And the endpoint they have put there is burning of sulfur fumes and then continue the heating for 3 days. That was their Shodhan method, intense heating. And then this particular 1 kg lot was distributed in 4 groups, so 250 grams of each. And then the first was treated with only Nimbu Swaras. 12 puttas were necessary to reach to that final Bhasma Pariksha. Then the second lot was treated with Gandhak and lemon. 11 puttas were required that time, Varaha putta. Then the third was Hingul and lemon, 9 puttas, and it was done. And the fourth was very interesting. I was very intrigued by that method, because in the fourth, this particular Shuddha Suvarnamakshik was treated with HGS, mercury and sulfur Kajjali form. So Hingul, Sinnabar is also HGS chemically. But here Kajjali is Shuddha Parad, which is processed in, using this garlic peels and rock salt triturated together. Then this detoxified mercury that is taken out. Then sulfur is processed with ghee and milk. Pouring is the process. That is triturated together maybe for 72 hours or 7 days. And then this particular drug is mixed with this Suvarnamakshik with lemon juice, and then they have used this Kupipakva method. Now, in this method, in Kupipakva is you have to heat it in a bottle using sand bath. The heating is for a lot of time, maybe 24 hours. Now, after the process is over, what you get inside the bottle is upper part, which is a different product, and lower part, they say they got Suvarnamakshik Bhasma. Further, that Suvarnamakshik Bhasma, they took out and they processed it for 6 times in the same Varaha putta, using lemon juice for trituration and sulfur. So to me, these are all 4 different products of named as Suvarnamakshik Bhasma. And if I am allowed to propose, there should be Suvarnamakshik Bhasma 1, Suvarnamakshik Bhasma 2, 3 and 4, because all 4 are different if you characterize them using XRD techniques. This particular group has gone ahead. They have studied Suvarnamakshik Bhasma in 2010. They have studied it for characterization in 2011 and 13, where they have studied it with same EDAX, TAME and XRD. And very interestingly, in 2015, the XRD comparison of different Suvarnamakshik Bhasmas, all these 4, all the 4, they have Fe, they all have sulfur, they all have copper, but the composition is different. And here, Dr. Kumbhar sir, we need your help. How the composition varies, why it varies, and whether this composition will give a different type of pharmacological effect.
Right. Sure, it will give a different kind of a pharmacological effect, and we surely can help you. Obviously, XRD is a good technique, but again, it is mainly a sort of a solid state analysis. It's basically the powder or what they take. What we would like to need is what will be the effect when you dissolve it in a medium, or maybe say an acidic medium. Let's take acid, which is easier to so we can. So maybe an atomic absorption would be good, but even further, maybe ICP-MS, which is the latest technique for trace analysis, can be tested on these in an acidic medium, and then their contents or constituents can be analyzed. So yes, that can be done, but pharmacological effects will surely be different. So yeah, we can help you with the analysis of some of those. for sure, and maybe a bit of a standardization if required can be done using ICP-MS or atomic absorption to further understand what would be the actual composition in aqueous medium or in acidic medium, which is easier because we need to dissolve that, and see what the effects may be.
So, so up till this point, there are two take-home points.
Sure.
That there has to be a standardization of raw material, and as far as possible, it should be the natural raw material as it comes, and there can be a separation of associated minerals, similar minerals. So identity and standardization of raw material is the first important step, and then the process standardization using, as far as possible, measurable instruments, equipments, and especially the temperature using muffle furnace. Yeah, there are a few papers which talk in favor of muffle furnace, and there are a few papers which talk against muffle furnace use. Again, for the sake of participants and my knowledge enhancement, if we calibrate, this paper has done that. If we do the process manually as described in the classics, say about 10 times, and we fix that temperature rising point and then coming down again, it's a parabola, or sometimes it's not parabola, it's high peak and then it comes down. It doesn't go like like this. So there is no this curve. It goes straight up and then comes down, but slowly. So there is a wide range you get. So if that is possible in muffle furnace, definitely replicability and reproducibility in the final. And from the research perspective, this was the second take home, that yes, manufacturing method, as far as possible, if we can keep it standard, then definitely errors will be reduced and we can get the drug which is reproducible. Or with the ingredients, the number, the amount, weighing, and the heat which is to be calculated. We did it for 2 of the bhasmas, and a few papers are there also. Kapardika bhasma this heat was calculated, and for Abrak bhasma this heat was calculated. So if we can come up with all these major important 20 drugs in Rasa Shastra, that to convert this drug from raw to bhasma, we need this much amount of heat in kilojoules. That would be another pointer where we can standardize the things.
Right. We very much do it.
Yes, I also.
So, yeah.
Thank you, sir and madam, for detailed discussion on the topic. You have already answered and addressed the queries asked in the chat box. And we have got many participants must have got ideas to proceed further in this regard in this topic. And we can go for the collaborative work, Ayurveda and chemistry. We can do collaborative work in Rasa Shastra. Thanks for this. Thanks for inputs, madam and sir. There were some questions in question answer section that they were asking. We have compared both the bhasma with these parameters, but do we need to go for clinical studies to compare the efficacy of these 2 types of bhasma, madam?
Yes, Vinay. Those who have asked the question, they are right. We should be able to do a clinical trial, but again, if we use the base of Ayurveda for clinical understanding, and then for that particular understanding we use some objective tests for obviously for Pandu, some of the liver function test and hemoglobin, or for cardiovascular, if we want to go to see the effect in Rudrog, then show us the imbalance in that particular disease, then and you give our bhasma in our particular following the Bheshaja Prayoga Vidhi at this time in this Prakriti, and it is possible, it's very much possible. So if we fit in that matrix well, then definitely we can show some evidence. But if we simply are going to go ahead with only whether this increases hemoglobin, it's not going to help Ayurveda, neither it's going to help modern science. So I think while going ahead with this, it is important. One more additional thing which Vinay, you have not asked and nobody has asked me, but I want to tell that there is a paper in Nanotechnology Science and Application Journal. It was published in 2021. This paper is by Rajendran Selva Kesavan. This person from Poland, and he is doing his PhD in Poland, and there is this Gregory Franklin who is his guide. They both are from Poland. They have come up with a wonderful review regarding Ayurvedic bhasmas as future nanomedicines. So everybody who has participated today, please read that. There are many, different language. The language is different than what we are used to. I understand that. But still, we have to know the language which world talks and world listens. And therefore, I think this will be a good paper. I'll just show you that somebody has said share the link. Let me try if I can share the link. Just a moment. Yes, I can. Copy. I will paste it in chat for panelist. Yes. Okay. So I did it easily today. Last time I was struggling with this copy and pasting. Yes, Vinay. So thank you very much.
Thank you,
I
madam.
also thank CCIH, Girish Tillu particularly. And I would like to ask Dr. Girish, do you want to ask any specific question?
Girish sir. Girish sir.
Ma'am, there is some technical glitch. I'll just fix it out from this.
Okay. So Kumbhar sir, we need your help
Yeah,
for characterization and analysis of the chemical nature.
Correct.
Nature and structure both, composition and structure both of this particular any bhasma which we will work with.
Right.
And secondly, again, for the participants, in between this characterization and clinical study, there is a area of pharmacological experiments. Now we are blessed that it has been proved n number of times that our drugs, if properly processed, they are non-toxic. In acute toxicity study, chronic toxicity study, they are proved non-toxic. But if we really want to come up with evidence, whichever drug you do, it's better, at least for the standard formulae which are used in the pharmacies and which are sold in all our shops, all the agencies which are selling all these bhasmas. If the formulation is standard, one method, and if we can do the characterization and toxicity study of that, at least those many drugs, those many final bhasmas, finished product, bhasmas as finished product, we can label as safe. And then there is no repetition of these toxicity studies. Then what will happen? Our energies will concentrate on seeing. There are many things which we need to see. We need to see how the bhasma enters into the body. So ADME, where it goes, whether it enters directly inside the cell or it goes through the interstitial spaces. Where exactly it reaches. So there are different screening methods available. And if we take the formulae which are prepared by our pharmaceutical industry, if there is a commonality, standard formulae, then definitely we can go ahead with these studies, which are still, nobody has yet started to check ADME of any Ayurvedic bhasma, which is very, very important. So ADME, then dynamics, and clinically we are working. So I'm not very much, I'm concerned, but I'm not deterred whether we should work on clinical trial or not. Yes, clinically we are using all these things. not as a single bhasma, but as in combination. But what we do not know is this in between area. And therefore, I think we should work on. Yes, Dr. Girish, sir, I'm seeing you. I just asked whether you want to ask some particular question.
No, thank you. Thank you, madam. This is very valuable discussion. I think Dr. Swapnil wished to say something. Can we make Dr. Swapnil as panelist? You want to share something as a pharmacologist, as you mentioned that ADME and how we can really do
BABE approach. He's asking about.
You want to add something? Over to you, Dr. Swapnil.
Thank you, sir. Namaste, Asmita ma'am and Kumbhar.
Namaste.
So I really like the discussion and interaction. So thank you so much.
Hello, Dr. Swapnil. Dr. Swapnil, you are visible.
Yes, he's joining back as a panelist.
Okay.
Hello, sorry, I think due to the transformation into the panelist, it took some time. So yeah, I totally agree with you all, ma'am. So with due respect, I just wanted to share that yes, we can focus more and more on many other aspects which yet to be discovered. But any new formulation or any manufacturer who is going to manufacture even the Schedule 3H drug, I mean the formulation which are already present in the Ayurvedic text, they also should establish their safety, toxicity, and PKPD data. Because even in the right now also in the market, if we go and collect 4 to 5 well-established marketed preparations, they may not show the similar physiochemical Rasaguna Vire Vipaka status exactly. And for that, I think our pharmacopoeia standards also need to increase, so that the way modern has called generic drugs or BA approach, the same kind of strict conditions and standardization and establishment of batch-to-batch reproducibility need to be there for all the drugs, not only for Ayurvedic proprietary medicine. So that's it from my side. So thank you so much, Girish sir, for giving the opportunity. Hand over to you, Asmita ma'am, sir, and Kumar sir.
Yes, Dr. Swapnil, I would take your comment very positively, but I would definitely like to tell you that if we when to do this, in case of Ayurvedic drugs, especially bhasmas, especially metal mineral drugs, they are in use for so many years, yet we do not have some data. That I agree. But you cannot compare them exactly with the modern drugs, because modern drugs have a definite molecular structure, and the generic is very, very standard. That generic molecule is used everywhere, right? That particular aspect is different than the Ayurveda aspect. And that I proposed is, at least let us see, I was talking about 4 end products, 4 bhasmas, which are different than one another. They're labeled as Swarnamakshik bhasma, but because the intermediate ingredients change, because the process changes, the end product structure and end product's composition does not remain exactly the same. Let us start with this safety, toxicity, PKPD, fine, but that does not that will not reflect in the present day existing manufacture of these bhasmas by Vaidyas for their patients and by the existing pharmacies which are there. If we want to do that, the whole practice will come in problem. I definitely do not advise that. I mean, I will not accept it at all. Not for a moment. You take it from me. So that practice will go on. That is our traditional system, and that practice has to be there.
Yeah.
The cautionary note is, yes, we are lacking in generating evidence for safety of our drugs to a particular extent. Just like any antibiotic, it gives you the side effects. It gives you this will happen if this is the dose. If you take it low, this will happen. that kind of literature we have to create. And therefore, to begin with, we should take a standard manufacturing process, which generally most of the 90% of the Ayurvedic pharmacies follow, to make maybe Suvarna Bhasma, Rajata Bhasma, Abrak Bhasma, Naga Bhasma, and let us standardize that one process, analyze it, do a PKPD of that, and then we can say that yes, we have generated the evidence for this particular Bhasma. I totally agree.
Ma'am, I think
I will not say that.
I think we both are in a synchrony,
On the same
absolutely 100% in agreement, and we both are saying the same, just in a different pattern. You are absolutely right as a clinician, and I'm also 100% in agreement with you. And Yuan, my observations, my understanding is very little, but what I can see, even as a patient, I trust more strongly on a formulation which my Vaidya has prepared for me, because he knows everything and he studies that very carefully. So I'm not against of that, but I'm very cautious about the pharmacist, well-established pharmacist. I will not take the names, but we all know who are the well-established pharmacists. And so for them, at least, it's feasible, doable, and logical.
Correct.
Because they are producing this on very large scale. They are supplying throughout. Now loan licensing and blah, blah, blah things are there. So in certain cases, for clinician, it's absolutely impossible whether he should treat the patient or he should do the research and evidence. That is absolutely, so all the clinician will throw this away. And there is no logic for the clinician asking to do clinicians, but all the established pharmacies who are selling the market products, even I see that when the good practitioners are there, Vaidyas, Ayurvedic Vaidyas, they say that you bring tablets of XYZ manufacturer, Asavarishta of XYZ manufacturer. This is because of their own experiences that the product of this is by this one only need to be procured. So why this is happening? Because although they use the exact same efficacy, safety, stability, and toxicity. So with respect to this aspect, all the pharmacies should be made it compulsory, and who are modifying anything in that process leading towards Ayurvedic proprietary medicines by adding anything new material in that, they should evaluate the Ayurvedic property of that new material. They should think about the fundamentals of Ayurveda as well as fundamentals of the modern science. At the time, start from the raw material collection to the finished product. So chemistry, biology, assays all need to go hand in hand and need to establish the trust the way modern science does. Although drug never understand whether it is modern or Ayurvedic. Chemical remains chemical, rasayan remains rasayan. How we look at it, that makes the matter. And I think that's how it all matters. Thank you, ma'am.
Okay. Yes, Swapnil, we are on the same page. But there is a lot to do in Rasa Shastra.
Yeah.
Absolutely. And people are repeatedly doing the same kind of things. And therefore, there is no new knowledge generation. So we need to look into it. I think at CCIH, we should start a collaborative lab, at least taking one drug. So bringing in one industry, we have a research lab and Dr. Kumbhar sir is there. At Bharti, we have pharmacology, wonderfully well set up department who is always working with us. So let's
definitely, ma'am. I look forward to explore the same.
Something.
Taking one case example
so,
and
what do
giving
you say?
Yeah.
Yeah, thank you, ma'am. So I think we have Dr. Kumbhar sir and his central instrumentation facility so that we can do so many things. And this is a call for all Rasa Shastra faculty and students. So any comments, sir?
No, I mean, I think we all agree to, come together and work. That's the best thing to begin with. And as Madam was saying, and as Swapnil was trying to put his point forward, supportive data, supportive scientific data, whether you use it or not, that's fine. But supportive data can be collected and be used as an evidence for the Ayurvedic Bhasmas at least. And I'm sure many things will come out of that. As Madam mentioned, the nano aspect is what people are reviewing. In fact, even in this Bhasma, if you see the procedure that they have used using Nimbu Ras and all that is an evidence that it is going towards the nano particle kind of synthesis. And that's what the observation is in terms of the size distribution. From 10 microns to 100 microns is what is the count they have found, both by using the furnace as well as the traditional method. So there may be some link can be found out by using all these methods that we have. So if anybody has this Bhasma ready, chalcopyrite or what we call as, we can start analyzing it even next week. We can give that as an experiment to the students. They will analyze it, and then we can take it up further and better the techniques. We can do that for sure. If someone has maybe a particular brand that they want to test, we are ready to do that.
Yes, sir. Thank you so much. I'm sure you will start about the Bhasmas also. We have 3 samples with you.
Sure. We will. Yeah, we are at your service.
Yes,
Thanks a lot, sir. Thanks a lot, sir.
Yeah.
For your critical comments, interactions, and discussions on this topic. Thanks, Dr. Swapnil Borse, sir, Asmita Vele, madam, for the discussion. Congratulations, Aarti Chandran. You have been awarded with this certificate for the presentation in JIM Club. We invite the participants. We invite the participation from PG students, researchers, faculty as a presenter for the next journal clubs. The information is there on this on the PPT. Please email your interest along with a statement of intent and 3 articles of your choice to editor.jim@gmail.com with the subject line as participation in JIM Club Journal Club. So that's all about today. I'm thankful to Professor Avinash Kumbhar sir, Professor Asmita Vele madam for their critical comments, discussions, interactions, and giving and telling us the opportunities in the Rasha Shastra and the collaborative work. Thanks, Dr. Swapnil Borse, for your critical comments, for your expert comments on this topic. I'm thankful to Dr. Aarti Chandran for the wonderful presentation and critical presentation on the topic. I'm thankful to Girish Telu sir and JIM Journal Club team, the technical team headed by Akash for their technical assistance, and thanks to all. Thanks. Thus, I declare that the session is over. Journal Club meeting is over. Thank you.
Thank you.
Thanks a lot.
Thank you.
Thank you.
Thank you, ma'am.
Yeah.